Hi everybody. So today I want to show you how you can do one of our lab activities of DNA extraction at home. So we are going to, in this activity, we are going to extract DNA from fruit. So what do you need? A fruit. I'm using here strawberries. See, I just need 2 strawberries to do this. You can use any fruit, anything actually that has DNA you could use. But you could make with bananas or blueberry or raspberry, really a tomato, anything that you may have at home that should have DNA. So if it is living, it should have DNA in it, right? You're going to need salt, just a little bit, 1 teaspoon of it, so you don't need the whole thing like that. You need 500 ml of water. You need 2 tablespoons of a dish detergent. Can be anything. It doesn't need to be transparent, clear like this one. It could be yellow, blue, doesn't matter. Okay, so just detergent. Alcohol. So I have here rubbing alcohol, isopropyl alcohol, and so you need some of this. I know this is probably the hardest ingredient to find these days. There hasn't been too much available. You're going to need a sieve. You're going to need a wide mouth cup. It doesn't matter how the cup looks like. It's just easy to sieve something into it. And you're going to need a narrow container. And look around your house. See what you can find. I have here like 3 different things, right? So champagne glass if you have one. So you could use that. So you see how it's narrow. You want narrow and transparent, okay? Here I have a travel size kind of little container of something. You see it's transparent and it's narrow. Also, you don't want it too short, okay? So I wouldn't make it shorter than this one. So it needs to be kind of, the tall is good. And I found this kind of container. It's like a candy container that I found. It's nice, it's cylindrical, it's narrow, and it's transparent. So I'm going to use this one for our activity today. And the reason why I chose to use this one and not the other ones is that the narrower, the better, because you use fewer ingredients, you use fewer materials. And if you use something too wide, wider than these things that I showed you, you're going to just waste a lot of material. And you don't need that, okay? So find something narrow, tallish, doesn't need super tall like this. It could be like a little shorter like this, but transparent so you can see what's happening in there. The first thing that we are going to do is actually get the alcohol and put inside of the freezer. You want it very, very cold for this. While your alcohol is cooling down in the freezer, we're going to prepare our DNA extracting solution. For that, you're going to need the soap, the dish detergent, the salt, water, some measuring spoons, and the recipe is you're going to use 2 tablespoons of dish detergent. You're going to use 1 teaspoon of salt and half a liter or 500 ml of water. Okay, so I'm going to kind of combine everything in this container right here. So let's start with the soap. So this is I only found a half tablespoon in my house, so I'm going to put 2 of these. So actually 4 of these, 2. So I want 2 tablespoons of soap. Okay, 1, so that's half. 1, 1.5, and 2. Now I'm going to use the water, which I already measured. This is there's 500 ml of water in there. I'm just going to use that to kind of rinse that tablespoon there. Okay. You're not going to need all this. It's just like, it's hard to measure if you kind of reduce too much. And then we're going to use 1 teaspoon of salt. Okay. The idea of this solution, now we're going to mix it. The idea of this solution here is we're putting soap. So it's pretty much the soap. The salt is kind of to balance the ions in there. But the soap is really what's going to work. And what soap is, if you guys remember, everything is made of cells, right? So our strawberries are made of cells, or our fruit, whatever fruit you're using. And the cells have a plasma membrane, and the plasma membrane is made of phospholipids. Now, phospholipids are lipids, like oil, right? So the soap here in this solution is going to break down that plasma membrane to kind of release the contents of the cell, and also break down the membrane of the nucleus, which is also made of phospholipids, to release the DNA that is inside of that. All right, now that we have our DNA extracting solution ready, we'll put this to the side because we won't need right away. We're going to get our strawberries and our Ziploc bag. Okay, so that's what you need. So you're going to put the strawberries inside of the Ziploc bag. You could kind of remove these leaves. Yeah, maybe better a little bit. And then we're going to put the strawberries, or whatever fruit you're using, inside of the Ziploc bag. So see that I'm using 2 strawberries. So if you're changing to a different fruit, you just kind of calculate the volume that you have, you know, and then make similar size to 2 strawberries. All right, so what we're going to do now is we close the Ziploc bag so nothing kind of leaks out of it. And what we are going to do next is we're going to smash these strawberries. Okay, so we're using your hand, you're going to just smash it. And what we're trying to do right now by smashing is we're trying to break the cells apart. Okay, so remember that this is a multicellular organ of the plant, the fruit. And whoops, my Ziploc bag is leaking. So I'm now going to turn it upside down. And so because this organ, this fruit is multicellular, they form tissues. And these tissues are kind of really pretty well attached together. Like these cells within a tissue are pretty well attached together. So what we are trying to do right now by this physical kind of smashing is just breaking those apart. If you're doing blueberries are a tough one because the skin is so tough. So you have to really, really smash it as well. Banana is really cool. It has a lot of DNA, but it's a little challenging because I don't know, it's just so thick to do it with the banana. And you can see, actually, you have this procedure in your lab manual, okay? So it's called DNA extraction. I believe it was activity 15 or 16. I'll get that corrected. All right, so smash well. Once you have kind of a pretty well smashed powder, what you're going to do is you're going to put some of this DNA extracting solution in it. Now don't pour, you're going to need maybe 3 tablespoons of it. Now remember mine is like a half, so I'm going to put 6 of this. 1, 4, 5, and 6. Okay, put this to the side. Once you put that DNA extracting solution in, you close the Ziploc bag well again. I changed the Ziploc bag because that one that I was using, I was being cheap. It was a reused one. I like to reuse my Ziploc bags. I wash them and then reuse, but it didn't work because it wasn't like sealing properly. So now we're going to kind of continue smashing this, and we want to make this solution as homogeneous as possible. So you don't want to see any, so you see, I don't know if you can see, but you can still see like quite a bit of pieces on mine. So I'm going to continue. And what we're doing now that we added the DNA extracting solution, what we're doing now is pretty much getting that soap, the detergent in the solution, to break apart the membranes, not only the plasma membrane, but also the membrane of the nucleus, so the DNA gets out of the fruit. So do this until you don't see pieces in, like, until you see almost no pieces in there. I think it's pretty good. I think I'm happy with this. I mean, if you're running, really honestly, if you're kind of, you could do this, like, let's say you blend some strawberries and you take the pulp out, you could do this with the pulp. Okay, and then just mix some of this extracting solution to the pulp, and then you can filter, which is our next step. All right, so our next step now is to filter this. Okay, so we're going to get a cup, and Move this out of the way. You're going to get a sieve. I usually do this with cheesecloth. It's actually the first time I'm going to do with a sieve, so let's hope this works. But it should work because what we're trying to do is just getting the big stuff out. The DNA should be going in this liquid that is kind of pouring to the bottom of my cup. Notice right now I'm using the big cup. So you're going to let this go in. You can kind of use a spoon and help it a little bit. Okay, so now the amount that you want will depend on whatever container you got to do the final part of the extraction. And because I got like a narrow one, I want to do in this kind of very narrow one so I don't use too much alcohol. Because, okay, the amount of alcohol, and I know right now with the coronavirus story, it's been hard to find alcohol. I wanted to buy more. I had a little bit in my house. I wanted to buy more for this experiment, but I couldn't find. So I want to use very little of it. Okay, so that's why like I'm going to use this very narrow tube because it will need like less volume. With it, you can actually kind of slide that sieve on the edge of the cup so you can get as much liquid as possible. And then what we're going to do, ooh, this is going to be tricky. I'm going to do on top of this. But what we need to do now is pour this juice, okay, so this fruit solution with the DNA into whatever your container is. And we are going to pour some juice to it, and we are going to put the same amount of cold alcohol on top. So you want to kind of put less than a half of your container. Okay, so I'm going to put like this, like a little between like maybe like 1/3, a little more than 1/3 of the container. Okay, I'm gonna put a little more. Okay, there. Okay, so see, I have now my juice here in my container, and what I need now is to get the alcohol from the freezer. All right, here is the alcohol. It's nice and cold. This is important, and what the cold alcohol is going to do is because it's cold, well, two things it does. Because alcohol, because of the charges in the alcohol, is going to kind of bring the DNA up, okay, out of this liquid here. And because it's cold, it's going to make the DNA kind of whitish, so we can see it better. Okay, now pay attention how you're going to do this. You're going to get your container and you're going to kind of lean it. And when you pour the alcohol, you want the alcohol to slide along the bottom part of this container, and slowly, because you want the alcohol to sit on top of this liquid. Okay, so don't shake it. You don't want it to mix. All right, it's not as hard as it sounds. Let's see. All right. Now you let it sit. Oh, it's already showing. It works. I'll show you in a little. And I'm going to bring the camera so you guys can see closer. Okay. I don't know how I can kind of focus on it, but I don't know. It's kind of also condensing. So let me kind of wipe this a little bit. But this pink thing, I don't know, can you guys see, like there's a pink thing that showed, well, it's pink because of strawberry, it's a strawberry, but it's really white, okay? So there's like a, I like to compare this as spit. It does look like spit, and it's kind of suspended in the alcohol. So that's it. So I want you guys to try this at home. If you have the materials, I think the most limiting right now is alcohol, but if you have this, it's super simple, super fun to do. You can do with many fruits if you want. And I want you to take a picture and send it to me. Okay.